Okay, are we ready to learn about things? We're gonna do, so the next 4 lectures are all cell cycle. I looked it over, I think we can do it all. Because it's about 6 50-minute lectures, so that should fit into 4 hour and 15-minute lectures. So first, exams are in, average was 75, which is awesome. I know 85 is even better, but an 85 average means I'm not making a challenging enough exam, and I'm giving way too much extra credit. So this is like a beautiful, nearly normal distribution. I mean, I'm impressed. You might notice there's some people who got in the 0 to 9. That's because some people were caught cheating. And I just thought I'd take a moment to say a few things about that. I can say that, like, and those people I believe are not even here, of course. But the thing that's upsetting to me is one person was shooting off my lectures that were in Blackboard on their phone. And I feel like I shouldn't have to police you guys. You're adults. And so now I'm going to have to police the final exam because now that I know that that's happening, it's just so insulting. Well, it's just really sad because I kind of feel like, what do you think you're going to do? Cheat your way into med school and then suddenly be smart? No. You're going to fail med school or whatever you want to do. It'll come out. So I don't know what people are trying to get out of it. So if you see me walking around doing like this during the final exam, it's not because I think you are cheating. It's because your classmates have ruined it for the rest of you. But anyways, the rest of you guys rocked it. So good job. Everyone's starting to come see me about what their grade should be if they want to get this and if they could skip the final exam and this and that. And if you go onto Blackboard and you look at your total points and you do that division and you got this many points out of this many possible points, if you're above a 91, you have an A. Or 91 or above, you have an A. And so please do not come take the final then. I'm not going to feel bad. Just don't take it. You're good. If you feel desperate and you need to see me, please come see me. Okay. And with that. Okay, so cell cycle. A lot of this is just intro-y stuff. Hopefully you all know it, so I'm going to kind of move quickly. So really the main purpose of the cell cycle is just to control copying the genome and then giving it to the daughter cells. That's the whole thing. And that has to happen correctly, so there are all these different points of regulation and these mechanisms for making sure the genome's copied correctly and it's given to daughter cells correctly. All right, so this is important for survival. If this goes wrong, then you don't have survival or you have cancer. It's all sorts of bad things. And it's needed for normal growth and to replace all the cells that die every day. So the major elements are chromosome replication. So that'd be S phase. So I'll talk about S phase over and over again. That's DNA replication, DNA synthesis. And then M phase, which is mitosis. Major point. Obviously you've got your G's in there, but the big things that are happening, copy your DNA and give it to your daughter cells. Right, so DNA is replicated towards the end of interphase. So some people got confused on the exam about what interphase was. That's basically everything but mitosis. So DNA is replicated towards the end of interphase, and if all goes well, so there's no errors, no stalled replication forks or anything like that, the cell will progress to M phase, where the duplicated chromosomes are segregated to the two daughter cells as a result of mitosis. So still a little break for me. Okay, so this is your classical figure that you've seen a million times of the cell cycle, which shows your gap phases that occur During? No, between S and M phases, right? So your G1 and your G2. So what's the purpose of these gap phases? Yep.
To make sure the conditions are okay for DNA replication.
Right, so before your G1 is make sure your conditions are okay, so environmental conditions. For DNA replication, what else are you looking for? So you need growth, right? So you need growth before, so in your G2 you'll have a lot of growth because you're about to divide, so your cell's going to have to become 2 cells, so it's going to expand in size. What else are you looking for? What do you not, or what are you not looking for? What would be really bad if you what do you not want in your DNA prior to S phase? Damage. Yeah, DNA damage. So you're looking for things like that. What about G0? We've learned about, I'm sure you've learned about G0 before. What's that? Yep.
It's outside of the cell cycle. The cell's not actively dividing at all anymore.
Right, so it's kind of like a holding pattern. There's no division, no nothing happening. Sometimes cells will go there if there's damage that needs to be repaired. Some cells, depending on the cell type, stay there forever. Good. Okay. So, of course, I'm going to talk about regulation because that's what we're all about in this class. So, cell cycles tightly regulated. It's controlled by a number of proteins, and they're acting as switches. And we talked already a few weeks ago. We talked a little bit about cyclins and CDKs and APC. And remember that paper we did in recitation? So that was your introduction. And so make sure you kind of think about that stuff a little bit now as we go into this. So these molecular switches, and they're not GTP binding proteins, other types of biochemical switches that allow the cell to progress into the next phase. And it's basically a kinase that gets turned on and phosphorylates things to turn them on or to turn them off or to target something to be degraded or something like that in order to allow that phase of the cell cycle to move forward. But you can only progress to the next phase if the previous phase was properly completed. So you can think of these, there are different points in the cell cycle that are called checkpoints. And these different points are present just to make sure things are going according to plan. So in late G1, you have your start, right? So is the environment favorable? Are you ready to go into S phase and replicate your DNA? You don't want to start replicating your DNA because that's starting the whole cycle if the environment is not favorable. You've got another checkpoint between G2 and M, and so that's prior to entering mitosis. And this is kind of looking at what's already happened and seeing if that happens okay. Did all the DNA replicate properly? Again, is the environment favorable? Because you just went through S phase, and if there's DNA damage, then you won't have appropriate DNA replication. You'll have stalled replication forks, things that are not good if you're going to try to segregate your chromosomes, right, in mitosis. So that's your G2M checkpoint. So you don't want to enter mitosis if things didn't occur properly in S phase. And then there's another checkpoint between metaphase and anaphase. What happens between metaphase and anaphase? Those who know their mitosis. I've seen people going like this. Right, you're doing this. It's an exercise. Chromosome segregation. Metaphase, remember metaphase plate. I always remember chromosomes line up on the metaphase plate. Anaphase is when you actually have segregation of your chromosomes, right? So it would make sense that there should be a checkpoint here because you only want chromosome segregation to occur if your chromosomes are appropriately attached to the mitotic spindle. Because if they're not appropriately attached, then you won't get equal chromosomes going to each daughter cell. And so those are your checkpoints. And so those are different points where cell cycle can arrest. So if a checkpoint is triggered, because something is wrong, then you won't move forward. Okay, so cell cycle is regulated. It's controlled by all these different proteins. Why is it so important to regulate the cell cycle? Why doesn't it just go? Why don't we just divide? Yes.
So you don't,
I just heard everyone say cancer over there. Yeah.
that's what I was gonna say.
You're gonna go cancer, ubiquitous. Okay. Yeah. Cancer, right? So obviously, if you have a lot of damage in your DNA, if you aren't attaching chromosomes to your mitotic spindle properly, all these things are going to basically result in either cell death or cancer if cell death doesn't work very well. Great. Okay. So I'm going to go through a few different ways that scientists study cell cycle. And so these are things to make sure you understand because, I love to have these applied questions on exams. So it's definitely important that you understand how you would study cell cycle so that you can answer those kinds of questions on the final exam. I just want to say that I'm angry at all of you for making me drink soda. I actually hate soda, but I drink it because I can't drink coffee or else I'll be up all night. So, I'm just kidding. I'm not really angry at any of you. I'm angry at myself. Okay. Okay. So all the machinery involved in cell cycle regulation is highly conserved. So this allows us to study a lot of the different processes in different model organisms. And so you may have seen these already in some of the papers that we've done. There's a huge amount of research on all the cell cycle proteins were all done in yeast, and that's why they're all called CDC something, because it's cell division cycle or something like that, and then a number, and that's because these scientists made all these mutations in proteins. There were just random mutations, and then they would look for phenotypes in yeast, and if they were cell cycle associated defects, distracting. If they were cell cycle, the people at home are like, what is she talking about? Dr. Ehrlich went mad. So if there was a defect in cell cycle, you can actually visualize some problem, right? If cell division's not happening properly, then you can see, the cells will look different compared to the wild type. And so that's where they were able to identify all these different CDC mutants, and then they characterized the functions of these mutants, and were able to identify all these different proteins that play a role in cell cycle. There's also xenopus animal embryos. So these are these frog embryos or frog eggs. Remember we did that paper just, 2 weeks ago where we are looking at APC, right? Does everyone remember that? And that was in xenopus. And then cultured mammalian cells. I think these are maybe HeLa cells. I forget. I found the picture. I don't remember what they were. So yeast is a commonly used model system because it's really cheap and it's easy to it's easy to grow, and it's easy to genetically manipulate. And so people either will use Schizosaccharomyces pombe, which is the fission yeast, or Saccharomyces cerevisiae, which is the budding yeast. And you can just see the differences. One divides by fission and the other one buds. So it's important to know this because if you're studying it and you're looking for a defect somewhere, you want to know what you're supposed to do. Okay, so here I'm introducing the concept of conditional mutants or temperature-sensitive mutants. And these are mutants that you can only see at certain temperatures. So you have a a permissive temperature and a restrictive temperature. And when you increase the growth conditions of your organism, in this case it's yeast, at your restrictive temperature, then you'll see some kind of defect. And this is usually because there's a mutation in a gene that results in a protein that isn't folded quite right, but at a normal temperature it's fine and it works fine, but if you increase the temperature, the protein misfolds just enough to not work. So the way people do this is they'll grow their cells at normal temperature, and they'll go through normal cell cycle, right? G1, S, G2, M. And then they'll shift the cells to a restrictive temperature where the protein whatever protein it is won't work. And then they'll look at the cells and see, where is this defect. So for example, here at the high temperature, what's happening to the cells? Are they going through the cell cycle normally? No. Would you say their growth is arrested? And where is their growth arrested? Between G1 and S, prior to the beginning of S phase. And so being able to see this somehow, you can start to think to yourself, okay, well, what genes are involved in entry into S phase? Oh, well, there are these different cyclins and different CDKs and all these different proteins that are involved, and that's how people did research. And so this is how a lot of these CDC genes were identified. So here's an example out of the textbook. So this is the budding yeast, and here they had this CDC15 mutant. And so wild type, which means at the permissive temperature where the yeast is acting wild type, you get the yeast looking, so remember this is a budding yeast, so it should make buds. You can see that the yeast is at different phases of the cell cycle, right? Because they're not all cycling at the same time. It's going to be a population cycling at different times. You could say that here it's just starting to bud. Here is a much larger bud. Right here, this one's not doing anything. Here it's starting to bud. So there's different things happening. So these cells are cycling. But here, what's going on? Something's going on here because the cells look like they're all doing the same thing, right? And so you can kind of think of it as like all the cells are running forward through their cycle, and then they hit a wall, and they all like pile up at that wall, and that's where the defect is at that phase of the cell cycle. So, what phase of the cell cycle do you think is being affected here? Huh? I'm hearing metaphase to anaphase transition. Oh, don't know about that. Telophase. Maybe telophase. Cytokinesis. Yep. Yep, because they're all just about to actually finish budding. But they're stuck. They're stuck. So what do you think CDC15 might do? Yep. Affect microtubules. I don't know. What do microtubules have to do with cytokinesis? Maybe affect actin? Yep.
I was going to say affect actin,
Yeah.
affecting the activation.
Okay, so maybe affect actin polymerization by affecting a nucleating protein. That's very specific, maybe possible. More specifically, did I look up what CDC15 does? I can't remember, but I believe it is a mitotic cyclin. No, I forget exactly what it is. Did I write it? No, I forget. So I'll have to look it up and see what it is. But yeah, so it's affecting cytokinesis, and it might be affecting any of those things. And then, so to make your brain explode a little bit, so you guys were just doing a great job integrating what we just learned into what we're learning now. And so we're going to learn about all these kinases that are specific for different phases of the cell cycle. So then we want to think about, well, what are their targets? What are they phosphorylating? Well, they're going to phosphorylate these kinds of things that will be activated and play a role in nucleating actin and organizing the contractile ring and organizing the mitotic spindle, all of these different things. Did your mind just explode? Just a little bit? Yeah, not really. Okay. Okay. Right, so these early embryos of all these different organisms are useful for studying cell cycle because they're very simplistic in terms of cell cycle, and they're very large, so they're easy to work with. So here you can take cytoplasm from a xenopus egg and then combine it with sperm nuclei and use it to study DNA replication. You can also study mitosis in cell-free system under control conditions. Do you guys remember where we recently read a paper which used this system? And that's, remember, where there are these mitotic extracts and these interphase extracts of these xenopus eggs. Yeah, so this is what they were doing. Okay, so then there's also immortalized cell lines. So these are just cell lines that we use in the lab. They can grow indefinitely, usually because something's wrong with them. Usually because they're cancer tumor cells of some sort, or they have some kind of viral gene. Actually, HeLa cells. You were in recitation today. So HPV, right? E6 and E7. That is what makes HeLa cells. And that's why who knows about HeLa cells? A lot of people, because you guys all know the immortal life of Henrietta Lacks and that whole story. Right, so it's E6 and E7 that allow these cells to reproduce indefinitely, and that's the same thing that killed this poor woman. So, cell lines, a lot of work is done in cell lines. A lot of the work I do in my lab is in cell lines. Okay, so how can we study? So these are different systems people work in, but what kind of assays do people do to study cell cycle? Well, you can just look at a cell and often know what phase of the cell cycle they're in, right? So if you look at these cells down here, what phase of the cell cycle are these? This one here, 1, 2, 3, 4, 5. What phase of the cell cycle are those on the periphery in? Interphase, right. And then what phase of the cell cycle is this one in? Yeah, I'm expecting that you just all know mitosis just like that, but. Well, so the things that you can look at are what's going on with your chromosomes and what's going on with your mitotic spindle, right? Because that green is your tubulin. So you've got condensed chromosomes, right? And then you only have one centrosome. Prophase. Right. Okay, so what about this one? That's an easy one, metaphase. Okay, so you guys are ready to go be cell biologists.
Let's do it.
Let's do it. This is good for the next 2 weeks.
Suck it up.
Okay, so you can visualize, you already know you can visualize DNA with specific stains, and then looking just at your microtubules, you also can learn a lot about where you are in your cell cycle. Is there a bunny? There's a bunny in the picture right here. Anyone who can see the bunny gets 10 points. Like here? Right there? Oh yeah. It is a little bunny. Okay. Good thing I'm not being observed. Okay. So cells in S phase. So that was really useful, right? So direct vision. Direct visualization is useful for studying mitosis. Cells in S phase can be identified by detecting incorporation of base analogs. What's a base analog? What's a base when we're thinking about S phase? I've heard someone say DNA. What's, yeah, so bases are they amino acids? Nucleotides, right. So a base, what's an analog?
Something that can change with it, but it has the same kind of structure.
Yeah, so it's different but similar. Right. So so you can use these base analogs. Specifically, one that's very commonly used is this bromodeoxyuridine. So it's a thymidine analog. So anywhere a T would be added, you would put this BRDU would be incorporated. And then you can detect them with antibodies that are against the BRDU. And so so this right here is showing a zebrafish gut. And they've got all their cells labeled red. So each little red spot is an individual cell. And then they've stained the cells, they've double stained, one with red for everything, and then BRDU, anti-BRDU is green. Okay? And so during DNA replication, remember you need nucleotides to be added in your new DNA. And so it's incorporated all of this BRDU in, and then you've treated your cells with an antibody that can recognize the BRDU and is fluorescent in a different color. So, what can you say about the green cells? What have they done recently? Yes.
They replicated their DNA.
Great, they recently replicated their DNA. What can you say about the red cells? What have they not done recently? I just crack myself up. Sometimes I feel like the exam questions I write are just as retarded. I mean, sorry, completely inappropriate use of a word. Just as challenged. So the red ones clearly have not, I'd never said that, it's on YouTube forever and I'll be arrested or something. So the red ones have not gone. The red ones have not gone through assays, right? So this is a very useful way for studying cell cycle in terms of whether cells recently gone through assays or not. Okay, yes?
So why would that show that those cells have recently replicated? I mean, wouldn't nucleotides, I guess, be incorporated in all the cells, even like just at random?
So when do nucleotides get added, go into your DNA?
It's only during replication, but not. Isn't there? But I'd be thinking like even if there was, well, I was under the assumption that even normal DNA in normal cells is always like having mutations and things.
So you're thinking of DNA damage and repair?
Yeah.
Okay, so your question is, how come you're not seeing also some green in the other cells because maybe they have damage that's being repaired? Well, so I guess if you were dealing with a whole person who's out in the world, in the environment, with all the different kind of exposures that we have, maybe you might see some sort of, if we could do this experiment on a whole person out in the world, you might see something, but don't forget this is in the laboratory condition, in cells that are not being treated with much of anything, well, it's a very controlled environment. And then also, if you're repairing a small stretch of DNA, and there's like 5 to 10 nucleotides that are being removed and added back in, and within that there's only 2 or 3 BRDUs added, the fluorescence is going to be much weaker than the fluorescence if a whole genome has been replicated. So I guess I mean, you're very right, but I think the intensity would be a lot weaker. Yeah.
So the ones that are yellow because they recently cleared the vision, those are no nucleus protect them from those tags from hitting the DNA? Or
No, so during S phase, you replicate your DNA, right? And so you as you're synthesizing your new daughter strands of DNA, you're incorporating new nucleotides, right?
Okay.
And every time a T needs to be added in, you have put all this BRDU into your cell, into your media, And so it can take BRDU instead. And so incorporates, and so it's an analog, so it fits and it can extend and actually be incorporated into the new DNA, but it's recognizable by this antibody, so you can detect it. Yep. Any questions about this concept? Okay, so researchers study, this is from the quiz this weekend, some of you may have gotten it. I had a huge pool of questions this weekend, so everyone got different questions. Researchers study cell cycle by adding the thymidine analog BRDU to cells. This is the type of question you might see on an exam. BRDU is incorporated into newly synthesized DNA. BRDU positive cells are likely to have, right. So I want you to be able to, this is like super basic, but it would probably be some like larger experimental things where you would still have to decide what phase of the cell cycle. So understand what BRDU is. Okay, so let's talk about flow cytometry for a minute. Who knows what flow cytometry is? No. Okay, that's fine. So basically, it's just a way to count cells. And it's a way to count cells. I feel like so many people are leaving and like coming and going today. It's so distracting. Sometimes, like, I feel like maybe they're handing out something free in the lobby.
Should we all go up?
I've seen like 3 or 4 people get up and come back, and I'm like, everyone has to be, I know. Okay. So flow cytometry, it's a way to count cells based on whatever attribute you assign to it. So let's say you have a population of cells and you want to count all the green ones. You tell this computer, count every single cell and tell me what percentage of them are green. Or you can do it based on if you have a mixed population of cells. Let's say it's cells from blood. Okay? So let's say I take your blood and I get rid of all the red blood cells because those are a pain in the butt. And all we're left with are your white blood cells. Is that a homogeneous population of cells, or are there many different kinds of cells? Different kinds of cells in the blood, many kinds of cells. Get rid of red blood cells. You've got your T cells, your B cells, your macrophages, your monocytes, your neutrophils, your eosinophils, your, I don't know, lots of, who's taking physiology? Don't they teach that kind of stuff? Yeah, a little bit. So all those cells have different shapes and different attributes, right? And so you can actually sort your cells based on how they scatter light. And so the way this machine works is very cool. It's a very cool tool. The way this works is you've got this machine and you pour your cells in solution. So you've got a tube that's like buffer plus cells, and the cells drip down. So each little red thing here is a cell, and lasers hit the cells. And the way the lasers scatter the light, it's recorded by a computer. Okay, and so you've got forward scatter and side scatter and whatever. Just like, so imagine, I'm a person. So like, I'm a person, and you're shining a flashlight at me. The way the light's going to scatter away from me is going to be different if I'm me or if I'm, the Jolly Green Giant, right? That's kind of a large individual. Right? Because we're different shapes, we're different sizes, and the light's going to scatter differently. And so the computer picks that up and can plot it on a graph. And so you can get these populations of cells that scatter light similarly. Or you can tell these lasers, hey, pick up all the green, or hey, pick up all the red, pick up all the orange, pick up all the blue, whatever. And so the lasers shoot at the cell, and it detects the color, and it sends it to a computer that says, there are this many cells that have been counted, this many are green, this many are this shape, this many are this shape, this many are this shape. Yes?
Color? Size or shape?
So that's what so it can be used for color, size, and shape. And so that's, yep. So I used to do a really nice paper in recitation, which did flow cytometry, but I don't know why, I must have stopped doing it for some reason. But usually what you do is you plot the cells based on two different things. So it might be forward scatter, which is a size and shape thing, because how you scatter light is based on size and shape alone. And then you might also do it based on color. And then it will detect it and it will plot it based on whatever you choose. It's really amazing. And so that's why I was talking about the mixed populations of cells. And so when people were originally doing HIV research in their earlier days, people would look at whole populations of blood coming out of cells coming out of patients and try to see, oh, these people have very low CD4 positive T cells, because you're able to use an antibody against CD4 and specifically identify those T cells, and then they're a different shape and size than, say, macrophages or monocytes or whatever. So it's pretty cool. So anyways.
Is this what happens when you get like a blood test and like
Yes. So when you get a blood test and they count cells based on certain things, they're using a version of flow cytometry. And so they're able to identify how many, if your white blood cells are when they say these kinds of things. Because then they count the total population, and there's a range of normal, and then they're able to say, oh, you have much higher than normal, you must be fighting an infection, that kind of thing. Yeah. So it's used in the clinic too. Were you going to ask a question?
Shouldn't they just like hook people up with like diseases or something like that, and just like filter their blood, like just it scatters this way, just destroy it type of?
So hook a person up to a flow cytometer and then say, hey, these people have this disease and it looks like this, and let's kill those cells. I like it.
that or the blood transfusion?
That is how they cured the HIV in those people, right? It was a bone marrow transplant where they like ablated their whole immune system and replaced it. Doesn't seem very cost effective, does it? Yeah. So let's see. So why am I telling you about this? Well, because this is useful. So let's come back. Okay. This is useful in studying cell cycle. I was coming back to it. So this is because you can actually stain your DNA and determine what phase of the cell cycle you're in by the quantity of DNA that you have in your cell, right? So if you think about the quantity of DNA in a cell through the cell cycle, is it the same the whole time or does it change? Okay, so at what point in the cell cycle does your quantity of DNA increase? S phase, right? Because you're copying your DNA, right? So you've got twice as much DNA in your cell at the end of the S phase. And when does that doubling of DNA go away? Huh? Mitosis. Right. So you got this buildup of double the DNA, and then the cells divide, and now you're back to single amount of DNA. 1x and 2x. So that's exactly what I'm showing you here, right? So you could stain your DNA with a DNA stain. Usually people use propidium iodide, which is an orange color. And then you put your cells in the flow cytometer, and you tell them, count the number of cells, count all the cells, and plot them based on their intensity of orange. Okay? If they're low intensity orange, plot them closer to 0. If they're high intensity orange, plot them closer to 2. So high intensity orange means more DNA, low intensity orange means less DNA. And so you end up with something like this, where you'll have a certain percentage of cells that are in G1. So they've just gone through mitosis. They have one copy of their They haven't replicated their DNA yet. So that's at the 1. And then you'll have a certain amount of cells that are in S phase. So their orange intensity is increasing, right? So this is this part over here. Now I want you to remember, this is number of cells. This is not orange intensity. People always get confused with that. Orange intensity is on the x-axis. So as you're moving away from 0, your orange is increasing. And then you'll have a certain populations of cells that have very, very intense orange. So they finish this phase and have not yet divided. So these are your G2 M phase cells. And then once they divide, they kind of end up in this G1 pile. Okay? And so you can use this tool to study how different things affect cell cycle. So if you're like, oh, I'm studying protein X and I think it affects blah, blah, blah in cell cycle. Well, you can actually do your experiment, whatever, add your protein X or your drug Y or whatever it is, and then stain your cells with propydium iodide, put it in your flow cytometer, it plots it, and you'll see where things are. And if there's some kind of defect in cell cycle, it'll look very different from this. You'll have your untreated and your treated, and you'll be comparing it to your control, and you'll see that something changed. Okay? So this is another way to look at it. Here, this is again propydium iodide staining, but it's just different. So you can either, with a flow cytometer, you can either show things in a graph that's plotted this way, or you can do more of a scatter plot, where each little dot is one cell, and it's showing PI staining. So this is the orange intensity from 0 to, in this case, 1023. And then here, it's they've decided to stain with an antibody that's against phosphohistone H3. That's a different fluorescent color. And it just so happens that phosphorylation of histone H3 correlates with chromosome condensation during mitosis and meiosis. And so the further out you are here, the more DNA you have, and the further you're up here, the more phosphorylated histone H3 you have. And so you can say that this population of cells is mitotic. Okay, so you have, so this is probably over here, your G1, here's your S phase in the middle, and then here is you're going into your mitosis, G2M, and then up here is your mitotic. Okay? And then just so you know, this is the orange I talk about. So these are just HeLa cells that are stained with PI and antitubulin, and that you can see their nuclei are bright orange. And so that's the color that's being detected by the flow cytometer. Okay? And so we're going to have a practice problem in a minute. So some of you may have gotten this in your quiz, and it may have been really confusing. So I made graphs. Okay. Wow, everyone's this very excited. That's good. Okay. So here's the test. So we're going to do this. We're going to do this all together. We're going to talk with each other and come up with an answer. So in order to study cell cycle regulation, researchers need tools to evaluate what phase of the cell cycle a cell is in. One tool, which I just told you about, is to stain the DNA in cells with PI and use flow cytometry to count the number of cells with different PI staining intensity. PI staining intensity will vary at different phases of the cell cycle due to differences in DNA content per cell. So you're working with a drug that should arrest the cell cycle prior to completion of M phase. And you expect the following results. So if you're working with a drug that you want to arrest your cells prior to completion of M phase, so metaphase, anaphase transition, you should have a picture in your head of what that graph should look like. And so you're supposed to kind of go through these and say control should look like this, treated cells should look like this, and this is how I would know that my cells are arrested prior to completion of M phase. And so I drew it out so that you guys can just kind of look. Let's see if I can do this. That. Oh. No. No. Oh, sorry guys. It's like no matter what I do. Okay, so you can't see D. Let's say. So it's A, B, C, D, and I have on the left control and on the right treated. So you've got your drug. And this is what you would expect to see if your drug was working, huh? Let's just skip D for now. It's not D. How's that? This makes things complicated. Okay, so I want you guys to talk to your neighbor for a minute. I'm sure this can probably go on, huh? It'd be nice if I learned how to use this. Better? I'll just stay away from it. Okay, so talk to your friends. You're looking, what are you looking for? You're looking for a drug that should arrest the cell cycle prior to completion of M phase. So at metaphase, anaphase transition. So you will not have cell division, okay? And so I want you to look, and so you have your control cells and your treated cells. You've got your cell number on the y-axis and your PI intensity on the x-axis. And so 100 would be lower intensity, 200 would be twice as high. Okay, so take a minute to talk to your friend and work through this, and I will come around and see if you need help.
Are these growing grass? Yeah. I was looking at it. If you look at this graph, this is, that's what it looks like.
The only reason why I would say A, because I've never been a fan of the treated one. But the treated Right, that's the reason why I say because the treated I'm not
It's
And then just thinking that higher one is lower. That's what it just is.
right.
In the middle, we started dividing as and then the last one is
You're right. Right, but didn't you say there's stuff in
There's stuff in everybody's
Right.
If it was split. I mean,
The first one didn't do that because he didn't understand the control. He just got it.
Okay, guys. Are we ready or are we eagerly talking about metaphase, anaphase transition? Eager. Okay, so did everyone understand the question? Okay. Does everyone understand how to read these graphs? Just remember, it's staining intensity. I just really want you guys to get this. Sorry. It's staining intensity this way. These numbers up here are cell numbers. So you put a population of cells in, they're going to be at all different places. So, 60% are here, 10% are here, whatever the leftover, 30% are here, right? And so you're comparing control and treated, because control is how your normal distribution should be. And then if you treat with a drug, it should affect something somehow. And if it doesn't affect anything, then, well. But so you're looking for cells that are blocked at the end of mitosis. And so which one here shows cells that are blocked at the end of mitosis when treated with drug? A. A. Did everyone find A? Does anyone have questions? Does anyone how about, okay, the A people over here. So tell me, why did you choose A?
Like you said, there was like a large population of the cells, and some of them would, since they like normal cells, would just go through the whole cell cycle. So some of them would be they would have more DNA in like S phase or whatever, and less in M phase, whatever. But the treated cells, they're stuck in that M phase, so like they still have all the DNA, they just haven't gone through. So most of them would have a higher intensity.
Right, so for the treated cells, exactly, since the block is at the end of mitosis, so you haven't had cytokinesis, you're going to get a buildup of cells with twice as much DNA. And so that's why you see this huge peak here at the 200. So what's happened is like these guys cycle all the way through, and then they hit this 200 mark and they stop, because that's where the block is.
So I'm not understanding why you have
more cells? I understand why you have more intensity, but why more cells if you did
Because
go through?
it's a population of cells. So think of it as like a bunch of like mice running through a maze. Okay? I know you're gonna think I'm crazy. Think of it as mice running through a maze. And they're just gonna keep running around and around, but they might be at all different places in the maze, right? But if you drop a gate down at one point, they're all gonna run and get stuck at one point. Okay? And so here you're just seeing they're all at different places, but here they kind of all get to this double DNA point and are stuck. They can't divide and go back to that 100 point. Okay? See me in my office hours if you're confused. Okay. Shall I move forward? Are we mostly satisfied? Do you have any outstanding questions? Okay. Go with it. No. Okay. Okay, was that fun? No. Okay. So that's the different ways people study cell cycle. Oh dear. Okay. So now we're going to talk about regulation of cell cycle control. Okay, so this depends on the activity of CDKs. Do you have any questions remaining over here? You guys are discussing? Come see me. Okay. CDKs, cyclin-dependent kinases. So people constantly get confused between cyclins and CDKs and what does what. Always remember CDKs are cyclin-dependent kinases. So they're kinases. They're the ones doing the phosphorylating. And they're dependent upon cyclin binding to be active. Okay, and that's why they're called CDKs. So these kinases control the activity of many different proteins that are involved in controlling the major events of the cell cycle. And they're activated by cyclin binding. So the cell uses the regulation of CDKs by cyclins to control the cell cycle. And it does this because the cyclin concentrations increase and then decrease at different phases of the cell cycle. When the cyclin is present, it'll bind to the appropriate CDK and activate that CDK, which will then phosphorylate things that are specific to that cell phase of the cell cycle. When it's time to exit that phase of the cell cycle, usually that cyclin will be degraded. And we talked about this in that paper where we were talking about APC. So you've got so I love this book because they make it simple. You've got your G1S CDK, your S CDK, your M CDK. Very simple. In reality, there's a lot of different numbers, but it can get confusing. Here it's just, so we're going to refer to it as the CDKs that are active during that phase. So G1S, S, G2M, and M CDKs. Okay? So you get this rise and fall of cyclins through the cell cycle, and they activate CDKs, and then they can go phosphorylate their target proteins. And so I really want you guys to understand this concept. Hopefully you did after that paper that we did in recitation. Does anyone have remaining thoughts, questions, and concerns about this concept? Okay, good. Okay, so I was thinking to myself, maybe, so people always say, well, what do the CDKs phosphorylate? And I was like, lots of things. And so I was trying to come up with a list of things that might mean something to you guys, because CDKs are active during different phases of the cell cycle, and so we've got these specific cell cycle specific targets. And so I was looking at papers for fun. This is what I do. And so I was looking for targets of CDK1, which is a mitotic, so it's an MCDK. And so there was actually a paper where people were trying to find targets of MCDK. And this is what, so this is just 40 of them that they've decided to highlight, but they actually found many more. And so if you look, and so you guys will all have this on your blackboard, but if you look at the targets of this mitotic CDK, there are lots of different things, but some things that are of interest is you've got proteins that are involved in actin polarization. So I see here GTPase activating protein for Rho1 and CDC42. Protein involved in Rho protein signal transduction. So right there you see you've got these GTPases that we just learned about that are involved in regulating actin polymerization and organization. Those are regulated by CDK, which kind of makes sense if you think about it, right? Spindle assembly, you've got these microtubule binding proteins, kinesin-like proteins, kinetochore proteins, all these different microtubule related proteins that are for spindle assembly. They are phosphorylated and regulated by CDKs. You've got some of these DNA replication proteins. You've got some other random things. They've got a DNA damage checkpoint protein. Makes sense, right? What else? Anything else fun here? APC activating factor. So basically, lots of targets, and if you think about it, it should kind of cut across a lot of what we've learned about. Let's turn this off. That's great. Okay. So lots of targets of these kinases. And so it's really important that these kinases are off when they should be off, because when they're on, they're going to set a whole lot of things, a whole lot of balls rolling. So there are many different levels of CDK regulation. And this is important, obviously, because if you don't regulate your CDKs properly, dysregulated cell cycle, not good, usually results in cancer. Or apoptosis if you're lucky. So these are just some different categories of processes that regulate CDKs, and we're going to go through them separately, but I just wanted to have this list. So the first thing is cyclin availability. If the cyclin is there, it'll bind to and activate the appropriate CDK. So that's one level of regulation, right? The other is availability of the target protein. Well, remember, CDKs are going to phosphorylate something. Well, if that protein's not there, that's another level of regulation. So then even if your CDK is active, if the protein that needs to be phosphorylated is not there, then you won't have activation. Then there's also inhibitory phosphorylation of CDKs. There are kinases that phosphorylate the active site of CDKs, and that keeps them inactive. And when they need to be activated, a phosphatase will remove that phosphate group. Then there's also activating phosphorylation. And so as we'll see in a minute, when the cyclin binds to CDK, there's a conformational change, which allows for a kinase to come in and phosphorylate the CDK in a different site, which activates the CDK. So that's another level of regulation. Without that phosphorylation, you don't have active CDK. And then there are these CDK inhibitor proteins, CKIs. And so that's cyclin kinase inhibitors. And so they bind and inhibit cyclin kinase complexes. Okay? And so you can have your cyclin CDK bound, but still kept inactive because they're bound by this inhibitor. So there's many levels of regulation, which kind of makes sense with such an important process. Okay, so what was I just talking about? I don't know. So for CDK activation, what did I just say? You need so here's your CDK, and it's normally inactive, and when your cyclin binds, you get this conformational change. So if you look at your red here, you're getting this T-loop reorganization happening, and that actually opens up your active site, and then this CDK activating kinase, CAK, will come and phosphorylate, and then you have this fully active cyclin CDK complex. So that's 2 points of regulation right there. You've got your cyclin binding and then your phosphorylation. And that's like your minimal requirements there. So then you can also have negative regulation of cyclin CDK complexes by inhibiting phosphorylation. So by inhibitory phosphorylation. Remember, I always say phosphorylation is not always activating, sometimes it's inhibitory, so it depends on where it is. And so here you can have your cyclin CDK, which has your activating phosphate, and then you can also have it phosphorylation by Wee1, and it'll be in the active site, so it will block the activity of the kinase, and so then it's inhibited. And so if you want to activate it, then you need CDC25 phosphatase to remove that inhibitory phosphate group. Okay? Sure. And so here are your CKIs, cyclin kinase inhibitors. And so you can have your cyclin CDK bound, and you've got your active phosphorylation, so it's all active, but then there'll be this CKI that can bind the whole thing, and it actually distorts the CDK active site, so it prevents it from functioning. And so, so you have this other mechanism of inhibition. So if you think about it, transcriptional regulation and cyclical proteolysis regulate the cell cycle, right? Because you're upregulating certain things when they're needed, so they can bind to things or phosphorylate things or dephosphorylate things. So there's this transcriptional upregulation. And then there's also cyclical proteolysis, so you degrade things that you no longer need. If you don't need your cyclin anymore, it gets degraded. You don't need the CKI anymore, it gets degraded. So what are some examples of some of the types of proteins that might be degraded based on what I just told you? What kinds of proteins would you degrade in this whole process, and why? So I hear you saying cyclins. Cyclins are the most obvious one, right? So cyclins would be degraded when? When would you choose to degrade your cyclin? Do you want to degrade your cyclin before it binds to CDKs? If you don't, so if you degrade your cyclin before it binds to CDKs, the CDK will never be active. So you don't want to do that. So maybe you might want to degrade it when you're turning off your CDK. So as you're exiting that phase of the cell cycle, you degrade your cyclin, and that shuts off that CDK. What other proteins might you degrade at certain times? So that's the most obvious one. How about CKIs? Right? So if you've got your cyclin CDK, that was this guy, right? If you've got your cyclin CDK complex that's being inhibited by a CKI such as P27, then a way to activate that cyclin CDK when it's time would be to degrade your P27. And that happens. Thoughts? No. How was everyone's Thanksgiving? I got a thumbs up in the back. Good. I ate a lot of food. A friend of mine posted on Facebook, what'd she say? Waistline reduction plan goes into effect tomorrow. I thought to myself, yes. She's a competitive dancer though, so I was like, of course it's on. Shut up. Okay. Anyways. Okay, so you might get degradation of things to turn things off, but then you also might need to upregulate certain things at different phases of the cell cycle. What kinds of proteins would you upregulate at what point? Cyclin, so there's another cyclin answer. When in doubt, cyclins. Right, cyclins, right? Because you need to upregulate, you need to increase expression of cyclins as you're going to enter a phase of the cell cycle so that you'll have enough cyclins present to bind to and activate your CDKs. And then what might you need to downregulate? don't know, maybe. We might need to regulate, I don't know, why did I put that there? I'm sure all sorts of things. Some of the target proteins, maybe. You might need to down-regulate at the end of certain phases of the cell cycle, maybe. Okay, we good? Nah. Okay, so I keep talking about degrading things. It's my favorite slides of the whole entire class. Right, so two different ubiquitin ligases that are involved in cell cycle regulation. One is APC, and you guys read about that in that paper that we did a few weeks back, right? And so here, APC degrades securin, geminin, as well as S and M cyclins. And so you're going to see coming up, we'll talk about it more, but securin is involved in inhibiting separase, which is the enzyme that cleaves the proteins that hold resistant chromatids together. That's securin. And then geminin is involved in inhibiting CDT1, which is involved in setting up your origin for beginning DNA replication. And so we'll talk about that more soon. But the way it works is you have your APC, and it's normally inactive, and there'll be an activating subunit. And once it's active, it'll target whatever its targets are for degradation. So regulation of APC activity is really important because it degrades some very critical proteins in the cell cycle. And if APC is active at the wrong time, that's not good, right? You don't want to degrade securin, when you're in S phase or when you're in the beginning of M phase. You don't want to degrade geminin at the end of S phase, which will make sense when you start to learn about it. You don't want to degrade your M cyclins in the beginning of M phase, otherwise you never go into M phase. So there's another layer of regulation here. It's not only your cyclins and your CDKs that are highly regulated, it's also your ubiquitin ligases. I'll introduce one more and then I will let you go. The other ubiquitin ligase are SCF ubiquitin ligases. SCF stands for skip colon F box. It's just the different components. And they recognize phosphorylated proteins. So one target are CKIs. These cyclin kinase inhibitors, when they need to be degraded, they'll be phosphorylated and they'll be recognized by the SCF, which will polyubiquinate and then degrade them. And so that will release the cyclin CDK. Okay, see you guys later.